hospital wards, and oil-polluted sites. That's Pseudomonas aeruginosa. Pseudomonas aeruginosa is known for its resilience and resistance. My name is Chioma Lillian Ozaduche. I'm here to take you to the microbial world where Pseudomonas aeruginosa is not just surviving, but potentially developing into something more dangerous. Most studies focus on hospital strains, but what about those we don't expect, those hiding in the environment, shaped by pollution and stress? We isolated Pseudomonas aeruginosa strain from a hydrocarbon polluted environment in Ejema Ebubu in Eleme local government area in Rivers State, Nigeria. This region had been known for producing crude oil since 1957. So we subjected the strain to whole genome sequencing after carrying out antibiotic non-susceptibility testing and also testing the impact of efflux pump. And then we asked, are these strains resistant to antibiotics? Do they carry virulence genes? How does pollution shape their evolution? How does this matter? It matters because Pseudomonas aeruginosa causes serious infection in immunocompromised people, and when it carries both resistance genes and virulence factor, then it becomes a real public health issue. We discovered that the isolates from this contaminated environment in Nigeria, one of the strains which we focused on, the one subjected to the host genome sequencing, was a multi-drug resistant bacteria. It was not susceptible to 3 different categories of antibiotics. The first, the beta-lactam, as in the case of kefepime. The second, the quinolone, as in the case of ciprofloxacin. And the third group, the chloramphenicol. But here is the kicker. These environments do not have high level of antibiotic resistance pressure, as in the case of the clinical setting. So why the resistance? There are 2 possible explanations for this. The first one is that hydrocarbons can select for efflux pumps or plasmid that carries antibiotic resistant genes. And the second is horizontal gene transfer, a situation of whereby the combined pollution from the heavy metals, hydrocarbons, and even possibly from antibiotics, they can drive stress response that promotes gene sharing. It was discovered also that from this study, because we compared this very isolate to other international isolates, worldwide isolates from the database that have their whole genome sequence data in the database, and this most of the resistant strain, they tend to have the most resistant strain have broad virulence factor profile, suggesting a link between increased antibiotic resistance and environmental stress from the hydrocarbon because of the overproduced efflux pump. So what does this tell us? Polluted environments might be silent incubators for antibiotic-resistant bacteria. And hydrocarbons, Yes, they contaminate the air, that's what we know, it pollutes the environment, but they can drive these bacteria to develop mechanisms, new mechanisms, to be able to gain resistance to antibiotics. From our study, we found out that there is an epidemiological relationship between the human strains and the environmental strain. For instance, the sequence type ST1503 assigned to the strain from the hydrocarbon area, the Pseudomonas aeruginosa from Nigeria, 1503. That same sequence type was also found in children in Mexico who developed bacteremia. That is, the sequence type was found from Pseudomonas aeruginosa strain that was cultured from children in Mexico that developed bacteremia, and also from cystic fibrosis patients in France. ST532 has been assigned and identified from oil wells and oil production water in Brazil. And the same sequence type 532 was also identified in clinical setting in Spain. ST918, this sequence type was identified in the respiratory tract samples from patients in a tertiary care unit in Germany. And this same sequence type 918 has been identified in Colombian oil residual water. ST267 from China, which has been identified in China, oil polluted soil and oil production field. This same sequence type has been identified in clinical setting in Tunisia, and also from drinking water in China. How about ST sequence type 24 for a global high-risk clone from China that has also been identified in both clinical and environmental setting? We can't just monitor hospitals. We need to look into the environment. We need to monitor what's going on in the environment, in water, in the soil, in industrial zones. Because in our war against antibiotic resistance, the smartest enemy might be underground at the place we least expect them. Environmental surveillance is very important. Environmental microbiology is public health. Thank you.